Afternoon, good morning, or good evening, wherever you are, and welcome to the webinar on training and orientation on validation of methods at the OECD. We are the 14th November 2023, and my name is Anne Gourmelon, and I will give you just one introductory slide about the objectives of this webinar, and then we will have our speaker for today, Miriam Jacobs. First, the objectives of this webinar are first to clarify terms used in the context of validation for test guidelines development at the OECD. Then another objective is to explain why validation of methods is important in the context of test guidelines development. Also, one objective is to guide laboratories embarking on methods validation on how to approach validation and evaluate method readiness, and how relevance and reproducibility of methods can be tested, evaluated. And then we will have a moment in this webinar for a Q&A session. So you are most welcome to use the Q&A box. We will be posting material resource in the chat, but the chat is not accessible to participants, only the Q&A box. So we will be selecting questions from the Q&A and raising them to our speaker and anyone who would like to answer. So also please note, this webinar is recorded and it will be made publicly available as a replay on the OECD website. So without further ado, I'm going to hand over the presenter rights to our speaker for today, Miriam Jacobs, who is the National Coordinator for the Test Guidelines Program in the United Kingdom. And Miriam, can you hear us?
Yes, I can hear you. Can you hear me?
Yes, very well.
Great. Okay, I'll start sharing my screen now.
Yeah, we can see your screen.
Yep. Okay. Okay, welcome everybody. This is really a preamble for the workshop that we will be having on the 14th and 15th of December later this year. And this is just to get common understanding with respect to validation terminology and some principles and considerations that are related to that. So I'm going to go to the next slide now. So here's an overview of the talk. Can you see my slides? Okay.
Yes, very well.
Yeah. Okay, great. So there are 5 aspects to this talk. The first part is to discuss why we want to have validation for regulatory purposes, why it's important. So the first part is looking at the crisis in reproducibility that we have and the relevance to the OECD test guideline program of the mutual acceptance of data principle. The second point will look at an overview of validation terminology, focusing particularly on prospective validation and more really the early stages of validation. The third aspect will look at examples of what can go wrong and why, and how we can recover from that. The fourth one will look at what you can do very early on in the process. And finally, we'll have some questions and answers, questions and discussion session. So the first one, crisis in reproducibility. That there's a lack of reproducibility in research is really very well known. There was a survey conducted by Nature and published in 2016 where they reported amongst their respondents, more than 70% of the researchers tried and failed to reproduce other scientists' experiments, and many couldn't reproduce their own experiments. And if you have a look here, unfortunately for us as working in toxicology and biology and chemistry, these were the worst failures really in chemistry and biology. So in biology, here you can see more than 60% failed to reproduce their own experiments. And this was from a sample of over 1,500 researchers, and it was just on the basis of a brief online questionnaire. But it's really quite shocking, and that really goes to show how one needs to be very careful with literature reports. Furthermore, in another report from Errington et al. In 2021, they looked at 193 experiments just to look at the protocol status, whether they were fully described and what the barriers were to conducting replications in the experiments reported. And in this report, you can see here that very often there was very little help from the authors when they tried to find out more about the protocols. In most cases, clarifications were needed. Often reagents were offered up to 50%, but often not. And then codes were sometimes shared, but often no response. And data sharing as well was really limited. Something that the JRC have reported on and given presentations on recently, this ProMap, Promoting Reusable Open Methods and Protocols. So there's a nice preprint from Sophia Batista-Liet online, where they give recommendations on how to report protocols. Failure in protocols and reproducibility in protocols is the first step in the validation process that's necessary for improving reproducibility of results and being able to transfer the test method. It fosters confidence in results that you can obtain with a certain method, and it really facilitates the use in the weight of evidence, hazard and risk assessment. And this is also supported by an OECD call for urgent mobilization of national and regional resources to support the validation of new methods for the safety assessment of chemicals, with a need to be able to have good validation and good information around first line protocols. The mutual acceptance of data and the practices regards the OECD test guidelines is the next key element to begin with that we need to consider. Reproducible data is essential to support the international mutual acceptance of data principle for all OECD member country chemical regulatory systems. It's critical for ensuring there are no trade barriers. It's critical for ensuring reproducibility of data, transfer of data, and the next few slides will go through these in more detail. The reason it was created was really in response to fraudulent studies submitted to regulators. The MAD framework was constructed to ensure the generation of high quality and reliable non-clinical test data for regulatory purposes. Good lab practice provides the quality standards, and the test guidelines provide the scientific standard. Regulatory authorities receiving the data under the Mutual Acceptance of Data Agreement under the auspices of the OECD know that particular quality and scientific standards have been followed. And they know then that they don't have to re-evaluate the test protocol to determine its robustness, as it has a consensus by countries via the test guideline program. And part of that approach really is truly by consensus. Every country, every member country needs to agree. So it's not by majority. What documents are covered by the mutual acceptance of data? The OECD test guidelines and the OECD principles of good lab practice are covered by MAD. Those are the only documents that are covered by MAD. All the other OECD documents are not. Guidance documents describing certain test methods in the series and testing and assessment aren't covered. Additional documents such as detailed review papers, validation reports, peer reviews, they're not covered by MAD. Guidance documents can often be validated test methods, but they are examples where the scientific standard wasn't agreed by consensus in the working group of national coordinators. And it's considered that the scientific standard for mutual acceptance of data hasn't been met at that time. But that doesn't mean to say that the guidance documents for test methods can't be adopted as test guidelines at a later date when more information and more relevant information is available to show reliability and reproducibility. What does the OECD Council Act on the Mutual Acceptance of Data say? It says that the data generated in the testing of chemicals in an OECD member country, in accordance with OECD test guidelines and OECD principles of good lab practice, shall be accepted in other member countries for purposes of assessment and other uses relating to the protection of man and the environment. However, the data requirements are government prerogatives. Interpretation of test results is a government prerogative. There is no need for any repeat testing for the same data requirement. However, acceptance doesn't automatically mean the use of the data by that country. The regulatory use, needs, data requirements are country prerogatives. The test guidelines are intended to address and respond to the existing or foreseeable regulatory needs in relation to hazard assessment. And the kind of regulatory needs that the test guidelines address, I'm sure you're all very familiar with these: determination of physicochemical properties, environmental fate and behavior properties, chemical mechanisms of action and bioactivity, absorption, distribution, metabolism and excretion data for model building, toxicity data for screening and priorities test setting, toxicity data for identification of a hazard or target organ, and toxicity data for characterizing a hazard, and residues chemistry data. Although data requirements are country prerogatives, and MAD is about avoiding repeat testing, if data requirements diverge extensively between countries, mutual acceptance of data principle will erode, as test guidelines data will not be accepted across countries having different requirements. Therefore, the more compatible and similar the data requirements are between countries, the more beneficial MAD will be globally. And although the interpretation of test results is a government prerogative, the test guidelines often integrate the transformation of the raw experimental data, for example, through prediction models, data interpretation procedures, to generate a test result that addresses more directly a regulatory need, for example, the identification of a hazard. The data transformation and interpretation procedures implemented in the test guideline are the outcome of countries' agreement to do so in order to generate meaningful data and reduce the room for misinterpretation and maintain a common level playing field. But it remains the member country's prerogative to use the standalone test result to satisfy a data requirement, or to use the test result with other sources of information in combination with other levels of interpretation or criteria that meet a country-specific regulatory need, and to not use that test result if their data requirement cannot be satisfied with that test result alone or in combination. So that's part one. regarding the crisis in reproducibility and the MAD principle, a mutual acceptance of data principle. Now I want to move to the section 2, giving a brief overview of validation terminology. Here I want to look at standard prospective validation, other aspects to consider, also highly relevant, but not going into any great detail here, are retrospective validation, test guideline revision, and test guideline augmentation. So retrospective validation, for example, can happen where if you have a lot of relevant data and you want to go provide a data package to show that the test method or improvements in a test method can be done on the basis of data collected historically. And this has happened several times, for example, for in vivo test guidelines. There are a couple of examples recently where we have done some retrospective validation. Test guideline revision also can happen with respect to more data being provided, being able to expand the chemical applicability domain, for example, or test guideline augmentation, when there might be a specific element of a test guideline that can be expanded further in a particular direction, and the evidence has been collected to be able to show how it can be expanded, or where a test guideline may have failed to become adopted and because it needs a bit of further work. And I will give one example of that later. But mainly the next few slides really focus on standard prospective pre-validation. And these are the main resources that we're using. So the OECD guidance document 34, published in 2005. And whilst it is being revised at the moment, and work is foreseen really for the next year or so, it's the basic requirements of validation. I don't imagine will change that much, not in relation to what we're considering here. And then the other resource is the paper from ECVAM, published in 2004, that was very much part of this guidance document 2005 at the time, a modular approach to the ECVAM principles on test validity. These are the modules. So starting first of all with a test definition, that is, what are the biological process and endpoints that are modeled by the test system? The second one is the within lab variability or reproducibility. Can the results be reproduced or replicated in-house by a different operator and over different time periods? Transferability. Can others run the method successfully? Is the protocol sufficiently detailed? Number 4 is the between lab variability. Can the results be reproduced or replicated by others? And how accurate is it? What is the sensitivity and specificity of the test method? Predictive capacity. Do the experimental results match with usually literature-based expectations, sometimes in vivo-based expectations, it varies, and broader scientific consensus? What is the chemical applicability domain? What can or can't be tested? What are the performance standards? What parameters can be developed that can be expected from this test method when it's working as expected based upon the historical data? And this includes reference chemicals to demonstrate equivalence in performance for new Me-Too test systems compared to those that have been previously validated. So these are different modules, and one can come in and out of the different modules. It's not rigid. And this is the from the Hartung et al. Paper, showing how they're constructed and how the modules lead when they're successful. They get discussed by the validation management group, and then they can be taken forward for peer review by various expert panels, wherever they might be, whether that's ESAC, the European ECVAM Scientific Advisory Committee, or ICFAM or GECFAM or OECD at this level. So really, I'm just going to talk a little bit more about these 4 modules. Reproducibility. A typical validation management structure would be having a study management team here at the top, independent chemical selection and expert review to the side, independent statistical analysis, and then a number of labs, at least 2, sometimes 3 and often more, it really depends. For the test definition, the objective is to define the scientific purpose, describe the mechanistic basis, characterize the cell line. There have been quite a few issues arising out of cell line characterization or lack of characterization that I will mention later. The limitations of the test method, the experimental design, and the optimized protocol, which includes the necessary SOP, the specifications of endpoints and endpoint measurements, and the derivation and expression of results and interpretation of a preliminary prediction model. The prediction model often needs to be revised on the subsequent steps that follow in the pre-validation aspect. And then there need to be adequate controls, vehicle controls, positive controls, negative controls. To begin with, also, one has requirements for the initial training or proficiency set of chemicals, and these need to be well-known chemicals to cover the relevant mechanism and mode of action of the test method and with negatives. With the second module, one starts to look at the variability over time and addressing variability for different operators, but using the same laboratory setup. And the proficiency set of chemicals generally expands. These are the most obvious chemicals due to quality and quantity of reference data, addressing the mode of action, the relative potency if possible, physicochemical properties and appropriate negative controls, availability, and representatives of relevant chemical classes for that particular test method. The SOP includes all of these aspects listed here, and it needs to be extremely well detailed. The first step with the protocol, with the transfer, is often the step where hiccups are found. Everybody tends to think that everybody else does the same sort of process and SOP and lab practice that they do, but it really isn't the case. So basic assumptions also need to be included, and these are the sorts of things that we've really learned over the years. It's much better to include a lot more detail than less to be able to ensure that the lab that the test method transfers to the lab successfully. For the module 3 with transferability, the robustness of a test, assessment of reproducibility of data in a second lab is assessed, and the practicability, the successful transfer. And this is an iterative process, and with amendments on the test definition and the protocol as needed. As the data is generated, problems arise, and as they're resolved, the clarifications need to be made. And for the proficiency set of chemicals, now as we move to this, they need to be well understood to cover the relevant modes of action. For the between lab variability, the objectives are the assessment of reproducibility of the experimental data in 2 to 4 labs and sometimes more. The training and proficiency set of chemicals need to be the most obvious chemicals again. Mode of action, potency, physicochemical properties, negative chemicals, and availability considerations. There's a slide coming up that gives a bit more of an overview, more detailed overview shortly. Here it is. So the essential elements for chemical selection are to address the sector diversity, to have a structural diversity, to have a good number of negatives, at least 25%, and ideally 50%. And it's actually this data that's often the hardest to find, the negative data, because it's often not published. Ideally, as we try to use NAMs or in vitro test methods more in IATA and defined approaches, the more we have information on the range of weak, moderate, and strong activity rather than yes/no response, the greater applications that we can use that test method for. But it's not always, it really depends on the quality of the data that's generated in the study. Sometimes it's just not possible to do that, not at the early stages anyway. Ideally, also you have human in vivo data, but obviously for some classes of chemicals, such as pesticides, it's generally quite unlikely to have this sort of concentration dose response information to a sufficient degree. Whereas one might have it for pharmaceuticals, the pharmaceuticals may not be wholly relevant to the wider chemical world that the test method is intended to be looking at. And then there are limitations and restrictions on international transport and use that we need to consider. Chemicals that are on the Stockholm POPs Convention that are banned, that shouldn't be really used anymore. They ought to be considered. Chemicals with abuse potential, it's very hard to import or export them in a validation study. So, for example, drugs, phenobarbital, cannabinoids, anabolic steroids, and so on. And then the availability, it can be very problematic to have a chemical that it's really rare and hard to find and there's no long-term manufacture, or if it's really expensive, it becomes really impractical. So these considerations need to be included in the chemical selection. There is no fixed number of chemicals. It really needs to be on a case-specific basis, depending on the literature and data that you have on which you can base your proposals for the chemical selection list. And it's often a very good idea to get that list that you derive, get it peer-reviewed independently and get buy-in as you go forward. And that's certainly something at the OECD level with the validation management groups and the expert groups. It's really good to have a good discussion with them, if possible at the outset, to resolve any issues that might come later. Whoops. Designs of a validation study. So what is not a good idea is to have loads of laboratories and just a handful of chemicals that are being tested, because then you're not really demonstrating the applicability, the chemical applicability of the assay. However, sometimes you don't really have a choice about the validation lab partners. If it depends really on the funding stream, but certainly, for example, if a CRO is interested in participating in a test method because they want to demonstrate, well, they want to gain skills in running that assay, and they will have a clientele that will want to use it. It's not really, I mean, if they want to participate, it's really to be encouraged. So sometimes one cannot define the number of or number of laboratories fully up front, but as long as you have a minimum of 3, it's encouraged in the expert groups. For past validation studies, for example, the situation on the left has often been the case where a number of different chemicals are run in all 3 labs, but it's also been commonly done to stagger or have phases in the validation study, so that you have your first core test chemical or proficiency chemicals in the labs that are run by everybody, and they all demonstrate that they can reproduce them well. And then you can expand your chemical applicability domain by testing in a number of different labs, or very often the lead lab will often test a lot more of the chemicals to be able to demonstrate the chemical applicability domain range. Independent statistical support is needed for within lab variability and between lab variability and predictive capacity. And the traditional process can take, well, this slide has been used, this interpretations of these slides have been used a lot to show the length of time that it can take, but sometimes it's been used to show that it can take a very long time, and that's really not necessarily the case. Research and development can take a great many years. The pre-validation process can include an awful lot of optimization of that research and development process, and sometimes it sort of goes backwards and you're really doing R&D. But on average, it can take 2 years, perhaps. Sometimes it will take a bit more, especially if there are problems arising. The validation stage, getting to full validation, where you start to have blind chemicals, which I'm not discussing in any detail in this one, that can take a year, or it can take a lot more. It really, it will really depend on a case-by-case basis. The peer review, independent peer review, can take a year, and then the OECD regulatory acceptance usually takes around 2 years, but it can take more for a number of different reasons, which we will come to in the next section. And then acceptance and writing into different regulatory jurisdictions often happens following the OECD regulatory acceptance. So the first stages here with the R&D can happen at the academic, industry side, or agency side as well. And then really, ideally, these validation steps, sometimes they are started independently or by industry groups or academic groups or so, but they can start to enter the OECD test guideline program at the pre-validation stage. And it's really good if projects do enter at that stage to get support from the international experts and expert nominations from the working group of national coordinators at that sort of stage. Because then you know you're going to be resolving problems that might come up later with respect to regulatory acceptance. You have a chance to be able to address those and get support. Intellectual property has become a bit of an issue also in the OECD test guideline program, as we have moved more to in vitro test methods. There often are intellectual property and patent issues around some of the test methods. It's not a problem at all now. And specific guidance has been developed regarding these elements in the OECD test guidelines. It's important to declare whether the test method contains protected elements right at the outset when the project is submitted to the OECD test guideline program. It needs to be explicitly indicated and the means foreseen to make these protected elements available to users if their method becomes a test guideline. So it's not a handicap, it just needs to be transparent. The test method needs to, ideally, it will address high priority needs for the test guideline program. And a guidance document for the development of OECD guidelines for the testing of chemicals from 2009 states that Only that the development or update of a test guideline is possible only when the proposed test method or modifications to the existing test guideline has undergone a critical appraisal concerning its validation and regulatory acceptance, including where feasible and relevant, an interlaboratory comparative study, i.e. a ring test or something like that, describing the performance of the test method. And then the current guidance document on validation from 2005 also states that test method validation is a process based upon scientifically sound principles by which the reliability and relevance of a particular test approach, method, or process is established for a specific purpose. So now, this is the one slide coming up, giving examples of what can go wrong and why. But this is what I will spend most of the time on. So some common stumbling blocks or hurdles in the OECD test guideline process. First of all, they can arise with reproducibility and transfer of the test method. And experimental corrections need to be conducted, and there may be missing characterization of the test method and its cell line. These have happened in quite a few examples of adopted test guidelines, as well as those test methods that have failed to become adopted. So, for example, one of the endocrine active test guidelines early on, the test method failed to be reproducible in another lab, even by the test method developers, and the cell line and the construct had to be changed. But then ultimately it ended up being a successfully adopted as a test guideline. It worked, it was fine. In another case, we've had a case of, very early on, we didn't do sufficient characterization of the in vitro test methods or the cell line, and we had an instance where another test method was found not to be the cell line that it was stated to be during the validation and upon adoption, and it was later found out by another group that the characterization was wrong and that cell line had been contaminated. The validation had been successful, the test method had been adopted as a test guideline, and a clarification sentence was provided within that test guideline. Sometimes the material supporting the performance or validity of the test method is not adequate. And so the greater detail given, the better, for all of the evaluations, the references, the validation reports, and making sure that they're all accessible. Sometimes there is a lack of adherence to validation criteria. Sometimes there can be issues regarding reference or proficiency chemicals. So here an example is the draft SIP induction test guideline, where although successfully validated by the JRC, the WNT wanted to see an expansion of the reference chemicals. So work on that is ongoing at the moment, but and it's resolvable. And there was a very strong justification for the chemicals that were used in the original validation, because they were the only chemicals that where human data was available. So this has slowed down the adoption, unfortunately, of this particular test guideline, but it's resolvable. Other considerations might be a poorly written draft test guideline, or sometimes, to be perfectly honest, the adopted test guideline, we find out later it's not sufficiently clear for the end users, and we need to go back and revise the data interpretation procedure. We need to clarify the language, but it's really only after trying it and having it out there that we are able to do those corrections. And we've had to do that a couple of times recently with the TG 456, for example. Sometimes there's a changed regulatory purpose of a test guideline, or it's not clearly defined. And increasingly, the WNT really does want to see a clear indication of the regulatory purpose of the test guideline and how it can be used, how it can fit, particularly now for in vitro test methods for NAMs. Sometimes there's a low level of commitment by the lead country. There can be a staff change or a reduction in resources, and the project needs to be taken over by another country, or it just gets put to one side for a year or two until someone is willing or able to take the project over, or it will get ultimately dropped if no one is willing to take it over. And you know, we all move jobs, resources change, so it's a realistic problem that occurs. Sometimes a project proposal is discussed at the working group of national coordinators, and further work is really needed to explore issues within that proposal. There might be a need for an expert consultation. There might be a need for a stronger evidence base for the proposal, and a detailed review paper might be requested first. And that has happened several times as well with the test method proposals going forward to the WNT. So we have that at the moment for just thinking of some with a MAPS test, for example. We've been doing a more detailed report and having an independent expert consultation for a project on the work plan. Sometimes things go awry because of how the expert groups are working. There can be insufficient briefing or management of or listening to new review experts or national coordinators and the interactions there, or the record keeping might not be sufficiently detailed enough. And so keeping really good records of all of the discussions and ensuring agreement on those records as one progresses really makes a difference in achieving efficiencies. And briefing new reviewers that may join, because often there can be circular discussions that start to occur as ideas or concerns expressed earlier and dealt with get reiterated by people who were not experts who were not involved in the work previously. Sometimes it's the national coordinators don't consider that the test method addresses a key endpoint gap, or there are a lack of lead countries coming forward to address an OECD identified gap. So, for example, with the thyroid validation effort at the moment ongoing, being led by the JRC, but with the discussions at the OECD expert group, countries do need to take a lead and come forward to address these assays. And these are assays that were first identified over 10 years ago in an OECD thyroid scoping document. So the delay there has really been a lack of countries coming forward to do the work. The JRC came forward and have done the preliminary or the pre-validation work for a large number of the assays, I think 17 or 18 or more. But still, more commitment is needed from lead countries to take forward some of these pieces of work. And some of them are. So, for example, PEPA are taking forward at least 2, I think, of the thyroid assays now. But more countries are needed to take these forward. Sometimes it's simply conflict of regulatory needs across different countries. And that means that the project won't be taken up. Sometimes also there can be cultural safety differences, or there can be issues around things that you think are easily available. So, for example, the human serum use and in vitro test guidelines, a UK project proposal that was made several years ago. A CRO came forward and was able to adapt some of the skin sensitization assays 442 for human serum use, and they had obtained the serum from Sigma. It was openly available. It appeared to be regulated, but actually it turned out not to be. And the issues that arose around the discussion at the W&T table really started to unpeel or expose a lot of different issues that we needed to consider when using human tissue, including competition with medical use, as well as exploitation of donors, for example. So that led to a much deeper piece of work, and including workshops to address those problems. So, and these things are not obvious when they first come. So, and they need to be resolved as we go through the process to be able to achieve full adoption as a test guideline under MAD. So there can be this, and this is just a start of a list really of really common, or they're not, I wouldn't say they're really common, but they really concrete examples of where there have been stumblings with respect to test method adoption at the OECD. Now, let's look at practical purposes, what can be done right to begin with. Really, it's really, if validation needs can be incorporated right at the outset into research outputs, the more we do that as we develop our work in the R&D side, as the test methods come through to optimization, the more that we have a detailed description of the SOP, the more we publish the data associated with the result, the more that the data is findable, accessible, interoperable, reusable, not as a PDF, but in a machine readable format, the more efficient the whole process will become, and the more transparent it will become, which will increase trust. There is insufficient space in scientific literature manuscripts for all of this sort of information, but there are websites where one can work on protocols together. There is, I mentioned right at the outset, this ProMap initiative and guidance developed by staff at the EUR-ELECFAM. This is the most critical step to really get on top of right to begin with respect to the smooth operation of validation. And the method transfer is often delayed at this step. And here are just a list of resources that one can go to improve the quality of the SOP and the data generated. So I've taken mainly European examples and OECD examples here, and there are a lot more internationally, but here is just a selection. The Pepper platform has particularly provided some very nice guidance on how to report test method readiness, for example. And there are a number of different guidance documents at the OECD. The GIVIMP is critical for test method developers. GD211 for describing non-guideline in vitro tests, and that's the basis of these test method readiness spreadsheets. Reusable and open protocols, and so on. So there's a whole list of resources here to make use of. And with that, I'll thank you for your attention, and we can have some discussion and questions now.